So thank you for the invitation today to the Crop Protection Network. We are going to talk about using remote sensing to understand an emerging disease that is affecting soybean. My name is Alejandro Rojas, and I am the principal investigator on this project. Most of the research that we are going to present today was done in the University of Arkansas by my student Qiurong, but we are now located in the Department of Plant, Soil, and Microbial Sciences at Michigan State. So taproot decline of soybean is a new disease, or sort of new, that is affecting soybean in the Southeast. This disease has been around for a long time, but we didn't actually had a name for it because we didn't know the causal agent. So we didn't know the pathogen that was actually causing the disease. And the reason for that is that the disease symptoms are very similar to other disorders or diseases that we observe. So in 2007, it was named as the mystery disease, and it was affecting states like Arkansas, Louisiana, and Mississippi. And the disease started moving to other states nearby, like Alabama and Tennessee. Finally, in 2017, we had a report that it was actually a Silarius species causing the disease on the soybean. And the first documented yield loss that you can also find in the Crop Protection Network was reported in 2018. And finally, in 2021, we now named the fungus, or researchers in Louisiana State named the fungus as Silaria necrophora. So this is the pathogen that is causing the taproot decline. And here in the map, you can see, thanks to Dr. Spurlock's research program, how many counties are being affected by taproot decline in Arkansas. So one of the problems that we had initially with this disease is that the symptoms are very similar to other diseases. It follows a template on soil-borne diseases similar to SDS, causing an intervenal necrosis and chlorosis that you can see here at the seedling stage and at later growth stages. But it's not just SDS that cause these symptoms. We can also see it with microphomina or charcoal rot. We can also see it with drought distress. We can also see it with a lot of different disorders and diseases that we observe in the field. But the name came from the damage that causes in the taproot. In this case, you can see the black stroma colonizing. So this black structure is actually the fungus covering the taproot of the soybean. And as soon as you try to pull out the plant from the ground, the taproot will break, and it shows how much damage has been done on the root by the pathogen. And the same happens at other growth stages. So it's very characteristic to see these signs of the black fungus colonizing those roots. But again, these symptoms can be really confused with other diseases, so can be confused with other diseases. So by the work that has been done in Louisiana and Mississippi, they were able to localize some of these species in this phylogenetic tree. So pretty much what they are doing here is looking at different regions of the DNA of this pathogen to try to understand where this pathogen is coming from. And it was associated to other Silaria species that are not really common infecting plants in the sense that they can cause disease, but there are a few examples of those that actually affected plants. But the other important factor that we get from this study is that there are different genes that we can use, or regions of DNA that we can use to identify this pathogen and actually use those for diagnostics. And that is part of the work that we are doing. So we are using some of those genes that help to build those relationships across the species to actually diagnose the pathogen and try to use this as a tool to understand the epidemiology. So in that great group that we have highlighted in the previous slide, there are other species like Silaria malai that cause root rot on apple trees, but it was a disease that happened in the US in the 50s, and just changing cultivar actually solved the issue. We also had Silaria arbuscula that was causing root rot and basal stem rot on sugarcane. So Silaria necrophora is sort of unique in the sense that it's not a common pathogen, or Silaria in general is not a common pathogen on agronomic crops, but it just happened to be in soybean, and we are trying to understand where the pathogen comes from. So this is a disease cycle that we put together. It's not complete. There is a lot of gaps that we have on this research, and that's the kind of questions that we are trying to answer. So we can start here when the fungus is colonizing some of the stubble or some of the crop debris that we have in the field. And that is where we see that the pathogen thrives. So it will produce this characteristic fruiting body that is called the dead man fingers, or some people will call it candle snuffs because of the tiny little threads that are formed by the pathogen, but those are going to bear different type of spores that the fungus can use to disperse. But we think that most of the colonization in soybean plants is being done by mycelia. So these are the threads that the fungus will use to grow on the plant and in the soil, and essentially those are going to extend and reach the root of seedlings. And this is the stage that we think that the pathogen is infecting the most. So we can see some of those symptoms that I showed you previously with the intervenal chlorosis and necrosis and the damage of the taproot. But essentially, the pathogen can colonize these plants and survive throughout the season, causing more damage as the crop develops. Going here on the right side, we know that those fruiting bodies, those structures that are produced by the fungus, they can harbor spores, but we don't have any indication that those spores are infecting the upper foliage and the stem of the plant. Rather, we think that some of those spores are going to colonize more dead plant material and keep thriving and producing more of the pathogen. But most of the inoculation or the initial inoculum that is causing the disease is happening at the beginning of the season that survive on crop debris. The other thing that is really interesting about Celularia is that it's a common genera of fungus that can survive in the forest. So other members of this group actually are really good saprobic fungi. And what it means is that they can grow really well in dead plant material. So most of the Silaria that you observe are probably present in the forest. So there is accumulation of leaf litter, and then you see these structures that will form finger-like stroma. And usually those are the ones that are called dead man fingers. But in our case, in the Salaria necrophora, we have those small or tiny fingers being produced on the stubble that is present in the field. So we know that it's a good saprobic fungi. We can also recover Salaria from dead plant material or infected plant material and get the pathogen in clean cultures. But we have a missing link in here because we also know by research in other genera of Silaria or other species of Silaria, but we are also missing a link here with the endophyte stage because we know that there are other species of Silaria that are really good on colonizing plant tissue and surviving as an endophyte. And what it means is that these fungus can colonize the plant and it won't cause any symptoms or any damage. So this is coming from research that it has been done mainly in forests and especially in the tropics, where we can have Silaria producing spores that are going to colonize leaf tissue, survive in the leaf, and then when the leaf falls on the ground, it will start colonizing again the litter and producing structures that will keep this cycle going. So we don't know actually how Silaria necrophora is surviving in the field in an endophytic stage. So how is Silaria moving from soybean to the next soybean season, or how is Silaria surviving in the field? So if we go to the basics, is the disease triangle. We understand, or we don't have a lot of knowledge actually, on how different cultivars of soybean are going to respond to this pathogen. There is some preliminary research done by other groups that show some cultivars that have some response. We don't understand really well the environmental variables that are driving the disease development in the field. So we don't understand the role of temperature, precipitation, and soil parameters. But we have anecdotal evidence that we have loamy soils that are actually good environments for these fungus to thrive. By the presence of the disease that it only happens in the southeast, we know that temperature could play an important role. We don't know what are the roles of environmental variables in the specific case of this pathogen. By the disease occurring in the southeast, we know that temperature and precipitation could play a role because these are very warm seasons and that can help for the pathogen to thrive and colonize more tissue. We don't have any evidence about soil parameters, but there is some anecdotal evidence that show that these pathogens tend to thrive in sandy loam soils. But we need to do a better job understanding what are the actual factors that are driving this disease. And in the side of the pathogen, we don't know what other hosts are actually being colonized by this pathogen. We don't know how the pathogen actually survives in the soil. Is just mycelia. There are specific type of spores that are going the pathogen to survive better on the specific conditions, and we don't understand a lot of the biology of this pathogen. So from that triangle, host is an important factor, and that is one of the questions that we have. And we were lucky that researchers at Louisiana State were already taking a look of how cultivars respond to the infection of Salmonella necrophora. So they tested the available cultivars to try to find some solutions for growers in a quick way so we can manage this emerging disease. And they were doing some inoculations in the greenhouse and trying to determine what cultivars were tolerant. Using this scale, based on the root reduction, they found that Osage, actually a cultivar that was developed in Arkansas, shows some tolerance to the pathogen. In this case, there was no reduction or less than 24% reduction of the root. So we are taking Osage as our tolerant cultivar and trying to understand how the pathogen causes disease in a cultivar that is tolerant and in a cultivar that is susceptible. But the other picture that we have is cover crops, and this is an important factor to consider because the cover crops are coming into the picture in agronomic crops. Growers keep adapting cover crops as a way to increase sustainability and increase the soil health. So over the last 5 years, we had an increase on the number of acres that are using cover crops. And one of the things that we have with cover crops is that they tend to promote soil health. So there are different types of cover crops. One is legume cover crops. These are going to be your red clover, your peas, and different beans that they have the ability to recruit different organisms that increase nitrogen in the soil, increasing your nitrogen credits. We have other types of cover crops that are non-legume, so these are going to be cereals and other broadleaf species that are going to have different effects on the soil, especially suppressing weeds and producing some chemicals that can control soil-borne pathogens. And the third option is the mixture of the legume and non-legume cover crops. But the whole idea with a cover crop is that we can grow these plants between seasons to do different things on the soil. First, increase organic matter, increase the number of nutrients that are present in the soil, and also maintain living roots in the soil that are going to increase the microbial diversity. And what we hope is that by doing those different activities in the soil, they are going to increase the soil health, and in turn, those effects are going to affect soil-borne pathogens. In this case, reducing the effect of the soil-borne pathogen and reducing the damage that is caused by the soil-borne pathogen. But there are different examples that show that some of these cover crops can actually have the opposite effect that is expected. So we have some examples with cereal rye where pythium can actually be increased and cause some damage on corn. There are examples of mustard that can actually be a reservoir for verticillium, causing more damage on the crop after the cover crop. And in the specific case of rye, we know that there are issues with planting, and in general, with cover crops, is a practice that causes some issues when we are looking at the different agronomic practices. But thinking about the biology with an emerging disease that we have, that is the taproot decline, we are asking the questions, can cover crops influence taproot decline? Is cover crop a good way to manage taproot decline? And the other question that we have overall, the big umbrella for this research, is how taproot decline behaves in the field over the season. So for that reason, my student Qiurong Fan is working on a project looking at some of these practices and actually using some novel technologies to understand how taproot decline actually happens in the field and how the disease progresses over time in a system in the field.
Thank you, Dr. Rojas, and I'll be taking over from here. So part of my PhD project is to understand the factors affecting TRD development in the field under different management practices. So we're using a multi-pronged approach to understand the epidemiology of this disease, which includes remote sensing, visual ratings, and diagnostics through qPCR assay. And so to investigate which type of the cover crop to be used in field, we conducted in vitro and growth chamber experiment. And here we are presenting the study done in a growth chamber using seedling cup assay. And we tested 9 different cover crop varieties that are commonly used in the United States, which includes wheat, barley, rye, black oat in the cereal cover crop family, and then vetch, winter pea, blue lupin in the legume cover crop, and then also the mustard, radish, and the brassica. We also added 2 varieties of the soybean, which is the Hutchinson and Osage. And each of these varieties of the seeds are placed into a cup filled with 2.5% inoculum, and then we would inoculate it with 2 isolates of the Taproot Decline pathogen, respectively, and then add a non-inoculated group as a control. So the cups were then incubated for 14 days before we collected the data, which includes the stem count, plant height, and plant weight, and then root weight and root mass, and also root DNA for verification of the pathogen presence. And so from the growth chamber seedling cup assay, we have analyzed the plant and root data. Here we're presenting the data of root weight reduction when comparing the inoculated group with the non-inoculated control. So here in this graph that each panel represents a variety of the cover crop or soybean. The x-axis is being the treatments that is either inoculated with the TRD isolate or MSU isolate. And our y-axis is being the percentage root weight reduction. We can see that the rye, barley, black oat, and wheat, these cover crops from the cereal category are more tolerant and more resilient under the pressure of the pathogen presence. With a good performance and in the seedling cup assay, and also being a popular cover crop variety in the US soybean cropping system. So we have chosen the rye to be the cover crop to be planted in our field study. So hence, in order to study the factors affecting the TRD development in the field, we artificially inoculated a field and then trying to assess the root and foliage health index to determine the disease progress across in different growth stage using the approaches that I mentioned earlier. And then we were trying to evaluate the cultivars with different tolerance level, and then to evaluate the impact of the cover crop on disease development. So here is an overview of our field study in which you can see that the field is divided into 2 parts. The top panel is planted with cover crop with a cultivar of the cereal rye Nan album. And then the lower panel was the fallow treatment, which followed the traditional tillage practice. So under each panel, the plots are a combination of either the infested or non-infested plots with one of the soybean varieties, in which the Osage were previously documented as the tolerant cultivar in a seed germination assay, and then the R10-30 being a susceptible, and we also adding a Hutchinson with unknown tolerance. So this field study was done in Arkansas that consists of 2 separate crop seasons. The cover crop season starts when the planting is usually happened in late October from the previous year. And then we would sample the soil from the field for analyzing the physical, chemical, and microbial properties of the soil. And then towards the end of the cover crop season, we would take the aerial images and sample the cover crop roots for assessing the growth condition of the cover crops. And then the termination was conducted in the mid-April by chemical killing. And the soybean season starts roughly around 2 to 3 weeks after the termination of the cover crop. The planting of the soybean and the inoculation of the pathogen happen at the same time. So at 4 different growth stages in this figure, you can see that's from vegetative stage 1 to 3, vegetative stage 5 to 7, and then reproductive stage 2 to 3, and reproductive stage 5 to 7. We would actually use the remote sensing approach, which is the taking the aerial images for analyzing the soybean vegetation index, and then root samples for quantifying the pathogen DNA using a qPCR assay. And then lastly is the visual disease assessment. Then with all these data combined together, we can track the disease progress in the field. The additional data that collected in this field study is the stand count at day 15 and day 30 post-planting and yield data at the harvest. And so here we wanted to show you the stand count data from our Taproot Decline field trial from this year. We have stand counts from day 15 and day 30 post-planting respectively. So we have cultivars that at different tolerance level at the x-axis, and then y-axis being the stand counts, and left panel being day 15, and here is the day 30. And within each figure that we have the plots that under fallow treatments on the left panel, and plots with the right cover crop on the right panel. So the brown box is actually indicated the inoculated plots, and blue is the non-inoculated plots. And in both counts, we can see that the inoculation reduced the germination of the soybean seeds by nearly 50% compared to the uninoculated plots in all these treatments. However, we can also see some of slightly reduction of the seedling at day 30 compared to day 15 in all these inoculated plots with the cover crop. And so here is another some background to guide us to better understand the remote sensing application in this study. The graph indicates the electromagnetic energy that radiated by the sun. So all this energy actually comes with different wavelengths. The visible light spectrum ranges from 380 nanometers to roughly around 700 nanometers, which can be seen by human eyes. And when this energy that it hits organism on the earth, they could distribute it in 3 ways. One is absorption, the second is transmission, and the third is reflection. And the energy that being reflected is the part that which can be detected by the sensors and being used for analyzing the object. So multispectral sensor usually captures a few bands in the visible spectrum and the near infrared spectrum as indicated in this figure. So why do we want to study these bands? Let's take this percentage reflectance of deciduous tree and coniferous tree as an example. So at all these different wavelengths that you can see the due to the difference of the pigment production in the tree, that the reflected energies that they varies. So we can use this variation to tell the differences between the vegetation. And let's see the figure on the right, which computes the normalized difference vegetation indexes. So, or people call it NDVI. And this ratio is actually calculated from the visible and near infrared light reflected by the vegetation. So the healthy vegetation on the left that absorbs most of the visible light when it hits it, and then reflected a large portion of the near infrared light. And unhealthy or sparse vegetation that on the right reflects more of the visible light and less near infrared light. So then we can see that with this formula that the healthy vegetation actually has a higher NDVI value compared to the unhealthy vegetation. So here are more examples of the vegetation indexes in formula correlated to relative traits. In this column of the formula, R represents red band, G is green, B blue, NIR is near infrared, and RE is the red edge. So multispectral sensor can capture the signals from all these 5 bands. So, for example, the NDVI we mentioned before that actually correlated to the traits that are usually chlorophyll content, biomass, or yield. And most of these traits that they can also represent nitrogen or they're related to nitrogen and other indexes that's correlated to the plant health. And so that's going back to our approach for using remote sensing to capture the field image. In our study, we use the strong DJI Matrix 200 and then carry a multispectral sensor. The first step was to plan a mission flight in which we define the area to be mapped in the field. So the flight path actually follows a zigzag patterns, and it's showing in the slide here. Here is our field. And we make sure that the picture was taken in a cross track and along track overlaps more than around 75%. And also to make sure the plant height and speed would actually generate a good spatial resolution to capture the details in each pixel. And it usually takes around 6 to 7 minutes to complete the flight. And the multispectral sensor that we use in our study captures images in 5 channels. And you can see each imager that has a corresponding bands at a certain wavelength. So each flight is capturing around 100 images per channel that would generate around 500 or over 500 images per flight for analysis. Actually, this is a relatively small data set comparing to some other breeding program or studies that using hyperspectral sensor that consists thousands of image or thousands of the bands per capture. And so upon finishing the image collection, the next step was actually to stitch these individual images into a reflectance map for each channel, just like indicated in here. And this step can be done in different software. And here we're using a software called Pix4D Mapper. And then the resulting field map is shown below. When we actually stack the blue, red, and green channel, it resembles the field we can see with our eyes, just like this. And so with the reflectance map that contains information in different bands, we can continue using the software I mentioned in the last slide or some other geographical information system applications to compute the index correspond to the relative traits using the formulas. In this NDVI field view, you can see that the area with healthy vegetation are showing green, and then no vegetation or unhealthy vegetation are showing red to yellow. And we can extract these index value from each individual plot and then process it to analyze the data for each plot and each treatment. Hence, we wanted to see the vegetation progress across the growth stage. And here is the normalized difference vegetation index. The plots treated with fallow are shown on the left, and then the plots with cover crop treatment are on the right. So we have this red curve indicates the inoculated plot, and the blue indicates the non-inoculated plots. And with the healthy foliage, that we can always see that NDVI from the non-inoculated plots are gradually increasing at a higher rate than the inoculated plots. We can also see that as plants growing from vegetative stage to reproductive stage, the canopy start filling and growing larger. The NDVI is slowly increasing some until R5 to R7, that the plant's growing into its peak maturity and then start senescing. So apart from the image analysis, we also conducted the visual disease assessment. Indices is the percentage of the plants showing symptom in the plot. And then the severity is the rates of the symptomatic plants showing disease at a scale from 0 to 5. As you can see, that the 0 being a very healthy and greenish, and then 5 being completely dead plants. We can then generate a normalized disease index using the formula provided, which is the severity divided by 5, which is the scale, and then times the incidence. And let's put the disease progress and vegetation progress across the growth stage altogether. There are 3 panels in the slides, with the top being normalized difference vegetation index, middle being incidence, and the disease severity being the bottom. So each panel has plots treated with fallow on the left and plot treated with cover crop on the right. When we rating the disease progress through the incidence, and we constantly observe that the plant death in the inoculated plots throughout the growing season, the right-treated plots has a higher percentage of the symptomatic plants and the dead plants when compared to the fallow-treated plots. So when we look at the severity plot, we can see that the inoculated plots, there are still some symptomatic plants presented in the field throughout the season. However, as plants growing into the reproductive stage, and these plants that they are slowly recovering itself from the infection. When we put in the NDVI and indices together, as incidence increases, the NDVI is decreasing since there's our ledge vegetation presented in the inoculated plots. Then we have to mention our results from the yield at the harvest, with the x-axis same being cultivar, y being the yield with a unit of bushel per acre. We can see that there is a higher yield in all the non-inoculated plots when compared to the inoculated plots. And interestingly, it seems that our plot with the planting of cover crop had a reduced yield when compared to the fallow-treated plots, regardless of the inoculation. So how does our remote sensing data correlate with the yield? We calculated the correlation between normalized difference vegetation index, which is the NDVI of the soybean, and then the yield. In this graph, x-axis being the yield, and then y-axis being the NDVI at different growth stage. This is the R2 and R3, and this is R5 to R7. It shows that the NDVI at both stages are positively correlated with the yield, and at R2 to R3 is with a coefficient at 0.92, and then it increases towards the end of season of at R5 to R7, which is 0.948. So it's seen that there is a positive correlation between NDVI and yield. And so next, we will have Dr. Rojas back in the session for continuing the talk.
Thank you, Fan, for explaining really well what we are doing. On top of that, we are tackling this question with multiple approaches. So we have the remote sensing, we have the classic plant pathology looking at disease symptoms and the incidence, but we also want to understand where the pathogen is happening, where is the pathogen localized in the plant? Because like with other diseases, we understand that these pathogens are localized at the root. So those symptoms that we see in the foliage often are caused by byproducts that are produced by the pathogen, like toxins that will move through the stem and go to the canopy and show those symptoms. So the other question that we have is, can we use diagnostics not only for diagnosing the disease, but also to understand the infection process of the pathogen? And here, we just want to explain the approach that we are taking. So Fan is working on developing a qPCR assay that is able to measure the pathogen from soil and plant tissue. And on top of that, we are going to have different internal controls that are going to help us to make sure that what we are detecting is actually a positive sample and not a off-target amplification. So what we are doing is, in a tube, we can measure with one color the pathogen, with another color the plant, and make sure that the pathogen is present. So this is your regular PCR or some of those diagnostic methods that have been used for other diseases that now we are familiar after a couple years. So soon we hope to have some data correlating to those three panels that Fan was showing in the previous slides and show how much pathogen is present in the root. So to summarize the data and the information that we presented today, Silaria necrophora is likely to infect different cover crops. Is more aggressive in certain cover crops, and all of this was done in artificial conditions. So we saw that most of those legumes are really susceptible to Silaria necrophora. We selected cedar rye for different reasons. One is one of the cover crops that is really easy to adapt by growers because it doesn't require any changes in machinery, can be planted really easy by existing infrastructure. Cedar rye can be planted really easy with the existing infrastructure that most growers have in the farm. So that is the reason that this is one of the cover crops being pushed to increase that kind of practice. But we also saw that it can still harbor some taproot decline. But so far, based on the effects that we see, we can say that it's neutral, it's not really negative, because we also know that there are some issues with adapting cover crops. And one of the problems that we saw in the germination or the stand count is that despite of being inoculated or non-inoculated, our stand count is lower than fallow. And the reason for that is that we need to do a better job making sure that the seed is in contact with the soil. And there is probably some competition or allelopathic effects that maybe are result of chemicals that are being produced by the plant compete or inhibit the soybean. But still, we need to keep doing some research on cover crops to understand how that practice is going to affect this disease and other soil-borne diseases. In reference to taproot decline, we are using alternative methods to understand the progression of soil-borne diseases. And you can see that remote sensing is a very valuable tool that can help us to understand what are the responses of different cultivars in the field. And we also need to combine that information with some of the traditional methods so we can do a better job characterizing the epidemiology of these pathogens. We also saw that the disease peaks at the seedling stage. So most of the infection will happen when these plants are growing, and it will cause most of the damage. Plants will grow and keep growing even if they're infected with taproot decline. We see that the severity decreases, and it's probably because of the canopy keeps growing, and that reduces the NDV or increases the values of the NDVI. So we have more green tissue, but still the pathogen is present. And that is the reason that we want to use the qPCR to monitor the pathogen over time, because maybe the pathogen colonizes and is not actually growing during the season, but it remains there causing some damage. Since the disease is happening at the seedling stage, seed treatments have been tested and show some effect on controlling the pathogen. Also, other methods of control, like soil applications in furrow applications, can be effective. And that is something that a collaborator in University of Arkansas is doing, Dr. Terry Spurlock. And he is showing that certain chemicals are actually controlling this disease by using soil applications. With that, I just want to acknowledge the group in my lab that really helped to put together all this project. I also want to acknowledge Dr. Terry Spurlock, that he has been a strong collaborator assessing these different aspects of the disease, and the people from the crop and soil environmental sciences that help us with doing the remote sensing and flying the drone for us, so FAN could help analyzing the data. I also want to acknowledge our funding sources, USB, Arkansas Soybean Promotion Board, and the Division of Agriculture of the University of Arkansas. And finally, I really want to acknowledge the opportunity to present today some of the research that we are doing. So thanks to the Crop Protection Network for giving us the opportunity to explain the research that we are doing and the results that we are seeing on Taproot Decline.
Thank you.